Hello to our friends joining us via recording. We are finishing up our discussion of lesson number 7. Lesson number 7 covers content related to the skin. So where we left off in class on Monday was looking at the 3 different kinds of skin cancers that you can develop. So who can remind me in the chat? Let's see if we remember. Which of these 3 kinds of skin cancer, so we'll call it number 1 for squamous, number 2 for basal cell, and number 3 for melanoma, which of these 3 kinds was the most common? 1, 2, or 3? Do you remember what the most common skin cancer is? Yeah, we're warming up the keyboard this morning, right? Yeah, so my most common kind of skin cancer is basal cell carcinoma. Number 2 in the way that I labeled them. So basal cell carcinoma, remember that kind of skin cancer forms when we have a cell that lives in stratum basal that becomes cancerous. Hey, remind me, when we were talking about cells becoming cancerous, it was because they were exposed to something. What was it that we talked about that melanin is supposed to protect those cells from, to try to help them not become cancerous? What was that problem that your skin gets exposed to? Yes, several of us frantically typing in, absolutely correct. When we talk about skin cancer, most likely where it came from is exposure to UV radiation. So if that UV radiation passed through some of my other layers, other strata of the skin, got down to one of my cells down here in stratum basal and mutated its DNA, turned it cancerous, that would lead to a basal cell carcinoma. Remember that when we talked about basal cell carcinomas, the good thing about them is that they grow down in this kind of uniform pattern, meaning that it's easier when we're trying to remove those cancer cells to get all of them. So because they're uniform and aligned, we can get them all when we take them out. That's not the case with squamous cell carcinoma and with melanoma as well. Remember with both of these that the cells grow down in an irregular pattern, and when they grow irregularly, they're harder to catch. I know it's long to type, so maybe you can abbreviate for me. When we talk about squamous cell carcinoma isn't from cells in stratum basal. Squamous cell carcinoma is from cells in which stratum? It's not basal. Where does squamous cell carcinoma start in? Do we remember? Yeah, some of us are ninjas on the keyboard. When we talk about squamous cell carcinoma, that's when the skin cancer comes from stratum spinosum. Remember from our lecture last time, stratum spinosum is cells that are alive. So if a UV radiation mutates one of these cells here, it will grow irregularly down. It grows a little bit faster than a basal cell carcinoma, and it grows in this irregular pattern that we see. But remember that the most deadly form of skin cancer is melanoma. And when we talk about melanoma, its name tells me what kind of cells have become cancerous. Which kind of cells are cancerous in melanoma? Do we remember who became a cancer cell? Who's going to build that tumor for me if I have melanoma? Yeah, so melanomas are tumors that typically are dark brown, or sometimes they're a little bit blue, purple, pink. The color's really weird because the kind of cells that are mutated are those melanocytes, the melanin producing cells. So remember that melanin gives you your skin color and your hair color and your eye color. Melanin's all over the place. So depending on the specific kind of melanocyte that is mutated to cause melanoma, that will give you these weird colors that we see. So this is where we ended up at the end of our class on Monday. What we didn't yet dive into, that I want to take some time now, is to talk about how melanoma has the ability to spread. So let me put in the chat for us here. When any kind of cancer spreads in the body, it's called metastasizing. So going from one place to another place. Oh, good clarifying question before we move on. The question was if when we're talking about melanoma, doesn't that one technically start in stratum basale as well? And that is correct. So melanocytes live in stratum basale just like some of those keratinocytes do. They live there together. But if I don't have any issues with the keratinocytes that are next door, it's just an issue with the melanocyte. That's how I developed that melanoma. But yeah, great clarifying question. Two kinds that start in stratum basale, one kind that starts up here in stratum spinosum. All right, so let's talk about metastasis or spreading when it comes to melanoma. So when I talk about melanoma, there are 5 stages, kind of. The first stage is stage 0, though. So on my scale of stages of melanoma, this is the same way that we talk about any kind of cancer, whether it's a breast cancer, a skin cancer, a lung cancer. We can always stage them based on how far they spread. So best case scenario, if someone has a melanoma, you catch it at stage 0. What stage 0 means is we are only in the epidermis. We have not spread to anywhere. Spreading is metastasis. So you're a stage 0 melanoma if you have not spread. Once you start spreading into different layers of the skin, so a stage 1 melanoma is in the papillary layer of the dermis, the upper layer of the dermis. If we spread past that papillary layer down into the reticular layer of the dermis, that's a stage 2 melanoma. Things get real dicey and really bad for us if we reach stage 3 of a melanoma. In a stage 3 melanoma, we've spread all the way down into the hypodermis layer of the skin. And what we find in the hypodermis are these things called lymph nodes. So lymph nodes are the things that your doctor will feel in your throat if you have a sore throat to see if they're swollen. Lymph nodes are connected to lymphatic vessels, which are kind of like the superhighways in your body. So if my melanocytes that are cancerous get into the lymph nodes, that's going to give them the ability to go anywhere in your body very quickly. So stage 3 melanoma, cancer cells in those lymph nodes. If we don't catch it in the lymph nodes, there's a very good chance it will change to a stage 4 melanoma. And in a stage 4 melanoma or a stage 4 of any kind of cancer, what leads us to call it that is that we find melanoma tumors in other places of the body. So maybe you start with a melanoma on your arm, and because it spread through the lymph nodes, you also end up with one on your leg or on your chest. Or maybe you end up with a tumor on your liver or a tumor in your lungs. The important thing about these liver and lung tumors here is if we were to remove them and take those cells out, those cells would actually still be full of melanin. When melanoma metastasizes, when it spreads, it's still melanocytes. It's still melanin producing cells. So even when it becomes cancerous, when it goes to other places in the body, it's going to keep making that melanin and just make a new tumor. So when we talk about any kind of cancer, but in this case, we're talking about melanoma, you can use how far it has spread to stage that melanoma. Yeah, so great connection. In the chat, there's the question, so this is kind of like our degrees of burns. Absolutely right. Yeah, the degree of burns was based on how far we had damage Same deal here with melanomas. It's how far things have traveled. How do we feel about our stages of melanoma? Thumbs up, or what questions do we have? Let me know in the chat. I'm getting some thumbs up. Dusty's good. Awesome. Lom's really good. All right. With that, we are done talking about the epidermis layer of the skin, the outer layer of the skin. Now we're going to transition into the dermis layer of the skin that's underneath it. When we talk about the difference between a stage 0 and a stage 1, it comes down to whether or not it's in the dermis. So now we're going to look at where a stage 1 or further melanoma would be inside of. First thing we need to know about the dermis is the place that the dermis meets the epidermis is not a straight line. It's not like the straight dermis and the straight epidermis right on top of each other. We saw this when we were looking at slides in the lab. The epidermis goes up and down and up and down, and it goes up and down because of the structure of the dermis underneath it. So in lab, I mentioned this terminology. Make sure you're familiar with it. Those places where the dermis reaches up are called the dermal papillae. The place where the epidermis reaches down are called the epidermal ridges. So we end up with this wavy pattern up and down that connects the epidermis to the dermis. That's good news for you because that's going to make it harder for the epidermis and the dermis to separate from each other. You can actually see the interaction between the epidermis and the dermis when you look at things called your friction ridges, or in normal person words, your fingerprints. So your fingerprints develop because of the shape of these dermal papillae. The way that your dermis is interacting with your epidermis is what gives you your fingerprint patterns. Fun fact about fingerprints is that literally no two people have the same fingerprints. Identical twins have the same DNA, absolutely, but their fingerprints are different because the structure of your fingerprint comes from the specific way that your dermis interacts with your epidermis. Scientists don't fully understand how the dermal papillae decide to organize themselves. It happens very early in fetal development. I think it's in the first trimester of fetal development that we start to see the dermis forming these patterns to interact with the epidermis. So no two people have the same fingerprints. And here's a question I need you all to help me out with. Your fingerprints don't change during your lifetime. Your fingerprints are the same. But when I think about my fingerprint, if I'm sticking my finger on ink and I'm putting it down somewhere, that ink is from the epidermis layer. And remember, we talked about how we're constantly losing cells from the epidermis layer. How is it possible, if these cells are always falling off, why doesn't your fingerprint change? If we're losing the epidermis all the time, why doesn't my fingerprint get lost with it? Any ideas? Yeah, so it really comes down to where the fingerprint actually comes from. The fingerprint actually comes from those ridges called the dermal papillae. Yeah, Shelby nailed it on the head. The dermis doesn't change. My epidermis, which is always interacting with my dermis, sure, my epidermis is going to change, but my dermis down below it is not going to change. So as long as my dermis is the same shape that it's always been, my epidermis will always grow back into the fingerprint pattern that I originally had. Now, one semester, I had this person who I think might have been a pyro. He just loved to talk about fire stuff. Um, so he asked if I wanted to burn off my fingerprints, what degree of burn would I need to do? What do we think? If we wanted to change our fingerprints, if we wanted to burn them off, what degree burn would I need to do to make that happen? Any ideas? First degree, second degree, third degree. What do we think? Yeah, so we're torn between second degree and third degree. Remember that none of us said this, but remember that a first degree burn just interacts with the epidermis. Epidermis is not where my fingerprints come from. A second degree burn gets into those dermal papillae, so we start to see some damage here, but I probably don't destroy the structures of the papillae. I might make them angry. I might fry some stuff. I like the way that Danae put it. Danae said, if you want to be safe, to make sure you burn that fingerprint off, totally right. We probably have to go down to a third degree burn, because remember, in a third degree burn, we get rid of the papillary layer of the dermis. It's destroyed. It's gone. When it grows back, it might grow back in a new pattern. So to be safe, we probably have to do a third degree burn. Second degree burn might, but what I suspect would happen is that we might get some scarred tissue, but still have our typical pattern. So I love it. Yeah, our true crime TV shows finally paying off, right? They tell us what degree burn we need. Liza's a little concerned about us thinking about those third degree burns, right? Please don't burn off your fingerprints. I promise it's not worth it. Just wear gloves, right? Not that I'm condoning crime, but just wear gloves. You're good. When we talk about the dermis, up here in the papillary layer, where we're going up and down, that is where your fingerprints come from. The other thing that the papillary layer and the reticular layer of the skin is important for is bringing the blood supply to the epidermis. So here's a word that we should be familiar with. This word is avascular. When I talk about the epidermis, I say that the epidermis is avascular, meaning it does not have blood vessels. But all of the cells in my body need blood, because blood's got two things in it that I need to survive. Can we remember in the chat, what are the two things that floats around in your body, inside your blood, that your cells need to be alive? Yeah, we're chiming in. The plasma that is floating around in your blood vessels, that's where we find our cell's favorite food, which is glucose. That's also how we get that oxygen transported around throughout the body. When I tell you that the epidermis is avascular, I'm telling you that by itself, the outer layer of the skin doesn't have any oxygen or any nutrients. The only way that any of the cells in the epidermis get their oxygen or their nutrients is because of these blood vessels here in the papillary layer. So we need these blood vessels up here in my up and down dermal papillae to make sure that my cells that live here in the epidermis have the oxygen and the nutrients that they need. The other thing that these blood vessels do is help us out with regulating our body temperature. When we talk about body temperature, this is something that is an example of homeostasis. So we will come back to here. I wanted to show you where we were going. Our blood vessels are going to help us out with the process of maintaining temperature balance. But let's take a step back and look specifically at homeostasis loops in general. There are many things in your body that we have to maintain at balance. So your body temperature, we typically run about 98.6 or 37 degrees Celsius. Can you think of any other things in your body that we might try to keep at some flat level? What other things in your body do you think we might want to make sure they don't change? And my wink, wink, nudge, nudge would be think about lab work you might have done. What are some of the things that your doctor would look at to see what the levels are? Okay, yep, so Hugh's talking about want to have the right amount of water. Absolutely, because we talked about hypertonic, hypotonic, right? So we want to have the right water balance. Yeah, Danae mentioned blood pressure. We want your blood pressure to be about 120/80. We don't want to be too high or too low. Yeah, your blood sugar level, your hormone levels, absolutely. Yeah, your salt levels, pretty much everything inside of you. There's a healthy range and there's an unhealthy too high or an unhealthy too low. To keep those things in your body at balance, we use homeostasis. So I'm typing that in the chat here. Homeostasis. Homeostasis is our balanced state. It's the normal that your body wants to be in. Because we don't live inside a little box where everything is always constantly the same, we encounter things that are called a stimulus. A stimulus is something that when you're exposed to it, it causes your balance to be off-centered. So this afternoon, when we go outside and it's like 80 degrees outside in March, okay, we go outside and our body temperature might go up a little bit because our outside environment is warmer. Or maybe we take that midterm exam and to reward ourselves, we go get some ice cream. I don't know. We eat something and our blood sugar level goes up. A stimulus is something that affects us and causes us to go into an imbalance. Because it's important that we stay balanced, our body has these things called receptors. All their job is to detect what's happening in your body. So when your temperature is flat at 98.6, they detect normal, they don't have to send any messages. When your body temperature gets a little bit too high, the receptor detects that you got a little bit too high, but my receptor has no idea what to do about it. That's above its pay grade. It can't figure that out. Okay, I detected heat. I need to send that information up to somewhere where it can be processed. So the control center is the part of a homeostasis arc that can figure out what needs to happen. When my receptor says I'm too hot, my control center has to figure out how to make me cooler, how to get me back to normal. When my receptor says my blood sugar is way too high, my control center needs to figure out how to fix it. So the control center is typically something in your brain or in the part down below it called the brain stem that figures out what needs to be done. It sends those direct So when I'm talking about information that my body is too hot, my control center would talk to effectors to cool me off. When I'm talking about my blood sugar being too high, my control center would send messages to endocrine organs that help me to bring that blood sugar back down. So everything in your body that we keep at a steady state, this is what our homeostasis loop looks like. A stimulus changes things, the receptor detects that things have changed, sends that information to the control center where it's processed, and an effector gets us back to normal. That's the general structure of a homeostasis loop. Let's apply this specifically to the process of thermoregulation. Let me type that word in the chat. Thermoregulation. thermoregulation, regulating your body temperature. So normal, my balanced state in the body, remember we said about 98.6 Fahrenheit or 37 degrees Celsius. I can get a temperature that is too high or a temperature that is too low. I'm pretty sure we've all heard this word before. What do we call it when our body temperature is too low? It's lower than it should be. This is what happens when we go outside in an ice storm, right? We might catch something. What might we catch? Exactly. Yeah, several of us are chiming in. If your body temperature gets too cold, it's called hypothermia. Hypothermia, just like we talked about hypotonic solutions that were less concentrated. When my temperature is lower than normal, that's called hypothermia. So let's take a guess then. If my temperature being too low is hypothermia, what would my temperature too high be? What would I call that if the temperature is too high? If it was hypothermia for low, exactly, hyperthermia when my temperature is too hot. So hyperthermia, I'm above normal. Hypothermia, I'm lower than normal. So let's start on this side of my loop here for hyperthermia. I'm too hot. So the stimulus is I live in Texas, right? I go outside in Texas in the summer. The stimulus is the heat from outside. My internal temperature rises. I'm no longer at 98.6. Now I'm at 99.6. That's detected by the receptors who are always checking my body temperature. There are two places in your body that are constantly checking your temperature to make sure it's the same. We have receptors that are in our skin, and we also have some receptors in the brain. The receptors in your brain are looking at the temperature of the blood that's going through your body. That blood temperature is actually higher than what we would measure. So when we take your temperature, we, like I mentioned, are measuring about 98.6. Your blood actually runs about a degree higher than that. So if we get even more than that, the cells in my brain, the cells in my skin detect that. Our body temperature is too high. Receptors detect that. They send information to the brain. The brain says, okay, we're too hot. I know how to fix this. We're going to talk to some effectors to help us fix this. The first effector we're going to talk to is the thing that we talked about in the skin that makes watery sweat. What was the name of the type of gland that made the watery sweat? And Wednesday lab friends, I hope we know this. Yeah, several of us chiming in. Excellent. The watery sweat glands are the eccrine glands. So the first thing that the control center, this place in your brain that only exists to regulate your temperature, first thing it does is it sends a message to the eccrine glands and says, hey y'all, we need to cool off. Eccrine glands kick into gear. So the eccrine glands start to spit out watery sweat. With that watery sweat goes heat. First response, when I'm too hot, my eccrine glands get activated. The second response, though, when I'm too hot, is I also talk to my blood vessels. I also talk to my effector, being the blood vessels. When you are too hot, let me get my pen, we take a blood vessel that's normally this size, and we turn it to this size. We do what's called vasodilation. I put that in the chat. Vasodilation. When I make my blood vessels wider. The more blood that I send to the skin, the more heat it's going to radiate off. So this is why after you exercise or when it's hot outside, your face gets flushed, it gets red. We're sending extra blood to the skin to try to radiate off that heat. So let's summarize what we've said so far. We went outside when it was hot. Our receptors detected that we're hot. The brain processed this information and said, I need to talk to my eccrine glands, my sweat glands, and my blood vessels. That's what we see over here, the blood vessels. I get back to normal. I get back to balance. I'm at homeostasis. I'm stable. But, 2 days later, because this is Texas, we have our next cold snap, right? We have our next ice storm. We go outside and it's cold outside. Our body temperature goes down. When our body temperature goes down, notice that the same receptors are used to detect that. My receptors are looking for normal, but it raises a red flag for them if I'm higher than normal or if I'm lower than normal. The flag has been raised, my temperature is too low. That information goes back to the same control center, the same processing hub. This time though, my processing hub says, oh, we're not too hot, we're too cold. When I'm too cold, I do not want to make watery sweat. That would make me even colder. I do not want to make my blood vessels wider and lose all of my heat out of my skin. Instead of activating my eccrine glands or telling my blood vessels to do vasodilation, I need to talk to a different set of effectors. So the first effector that I talk to when I have hypothermia, when I'm too cold, is a type of muscle tissue that attaches to your skeleton, helps you to move. Which kind of muscle tissue? Well, we also learned, right, that it generates heat. It attaches to the skeleton, helps you to move. What muscle tissue am I going to activate? Yeah, so several of us are mentioning I could at first, I would activate that erector pili muscle to try to give me goosebumps. But remember from lab, goosebumps alone aren't going to actually make us warm because we're not animals with enough hair. We'll actually go one step above goosebumps and we'll start to shiver. When you're shivering, it's your skeletal muscle tissue that's contracting. Shivering moves your whole body, right? Everything starts to shake. The goal of everything shaking is to generate some heat because the process of muscle contraction creates heat. So if I'm feeling cold, my control center in my brain will send a message to my skeletal muscles to start shaking. I'm going to shiver. The other thing that my control center is going to say is, hey, blood vessels, you're normally this size and I lose heat when you're that size. You need to get smaller. You need to do vasoconstriction. Vasoconstriction. So my blood vessels that I see by my skin are going to squeeze smaller because any blood that's up by the surface of your skin is losing its heat. It's cooling you off. So to save me from losing a bunch of heat, I'm going to make those blood vessels smaller. This is why when people talk about having hypothermia, they talk about having like blue lips or your fingers getting blue. That's because the blood vessels in those places have shrunk really small. We're not sending as much oxygen-rich blood to those places because instead of sending it all to the skin to radiate off heat, we're actually sending it all to your core, to your thoracic cavity and your abdominal pelvic cavity, where we've got all those important organs, right? Organs, here, let's make a big picture connection. Organs really care about us being at temperature balance. They really care about us being at the right temperature because for them to work, they need to have their enzymes not get denatured. When I talk about a protein that is denatured, what's going on with that? What's happening with denatured proteins? Dr. Ollis either has a really bad lag or everyone's sleeping. Perfect. Yeah, so the first thing that happens with the denatured protein, it loses its shape. One of the ways that they can lose their shape is if they get too hot or they get too cold. So if my body temperature is too hot or too cold, I can wreck the shape of my proteins. And remember, if I lose my shape, I also lose my function. So when your body is trying to decide, should I keep blood flow out on the skin or should I pull it into the organs? Your body's thinking, you know, it would be really bad if the enzymes in my liver couldn't do their job. Send all that blood to the liver, to the abdominal pelvic cavity, to make sure that my proteins can keep doing their job. So vasoconstriction, we shrink down the skin blood vessels to make sure that we don't lose heat there and we save that heat in your core. Yeah, I love the way that Hugh summed it up for us, reminding us of day one in class, right? Shape determines function. If I get so cold I lose my shape, I'm going to lose my function as well. And the same is true, by the way, if I get too hot. If I get too hot, I can mess up those proteins too, and they can't do their job. That's why it's critically important that I be able to do thermoregulation. Thermoregulation, that's what we're talking about here. Having the right temperature balance. There's a question in the chat about if you're anemic. So the level of red blood cells in your body is another thing that we could make an arc like this for. So if you're anemic, there's a certain level of iron you're supposed to have in your blood and your red blood cells. If I have a really iron-rich meal, I can go through this process where a receptor detects that. My control center says, hey, let's make some more red blood cells. And that'll cause my bone marrow to make me more red blood cells to get me to normal. Or the opposite could happen as well. If I am anemic, I don't have enough, my body is either going to try to steal iron from other places, or I'm going to start to crave foods that have iron in them to hopefully get me back up. So anemia is not exactly what we see on this chart, but it's another thing that's regulated just like this. Same thing with blood sugar, same thing with a lot of those balance things that we talked about. If we need to have a normal range, we've got these kinds of mechanisms in place to help me if I'm too high or if I'm too low. What other questions do we have? Dusty, I'm not totally familiar with Raynaud's syndrome. What's happening in Raynaud's syndrome? I suspect that and pretty much any other disorder, what's happening is I'm out of balance. Somehow my control center is not sending out enough of a signal. Okay. So for some reason, the fingertips are losing their blood supply. Yeah, so it could be either that the control center thinks that we're too cold, or it could be an issue. Now that you mention what's happening, I feel like I've heard a little bit about this. I think that's actually more of an issue with blood pressure balance. So one of the ways that you maintain your blood pressure in your body is you expand blood vessels or contract them. And if we're doing a bad job maintaining our balance, we might make things smaller than they're supposed to be. So if I make my blood vessels in my fingers smaller than they're supposed to be, I don't have blood flow there, and my blood pressure is much higher there than it is in the rest of my body. So I can't tell you that 100% is the reason, but that's what I suspect, that we've got an issue with how constricted or dilated the blood vessels are. This system is not working as it was intended to work. Great question. How do we feel about thermoregulation? Thumbs up, thumbs down. What do we think? Good. Getting some thumbs up, getting some smiles. Perfect. Hey, I'll mention that next week, we're going to look at this same kind of chart that has to do with blood calcium balance. Because one of the ways that your body maintains blood calcium is by building bone or breaking bone. So coming soon, we will see this kind of thing again. This is what we call, let me see, okay, this is what we call a negative feedback loop. Negative feedback loop. And so what I mean by that is when I detect something, so my stimulus made me colder, my temperature went down. My effector is going to do the opposite. So my effector makes my body temperature go back up. Or if my stimulus makes my body temperature go up, my effector is going to make the body temperature go down. So in a negative feedback loop, the feedback in my system is always going to be the opposite of what I do to address it. Negative feedback loops. I mention that because there is one other kind of feedback loop that we have in the body that we need to be familiar with. And these are called positive feedback loops. In a positive feedback loop, what my effector does, the change that happens, I actually do the same thing as was my stimulus. So a good example of this is how we form blood clots. So say we get a paper cut and one of the blood vessels in that papillary layer of my dermis, one of those blood vessels gets ruptured. When blood vessels get ruptured or when they get damaged, this causes my platelets, those little tiny things on the blood smear slide, to spit out some proteins. These proteins, a lot of them will form into big, long fibrin proteins to build a patch. But the other thing that happens when I spit out these proteins is it sends a message to the platelets that are around me. Oh my gosh, we need to freak out. We need to make a blood clot. So more platelets come over, they spit out their chemicals. We're making more proteins. But as they spit out their chemicals, more platelets come over, which makes them spit out their chemicals. And that recruits over more and more. In a positive feedback loop, my original stimulus is made even more in this loop. So this will go and go and go until we've built the blood clot. So the way I like to think about a positive feedback loop is instead of us immediately going like we did with the other one, where if I got too hot, I'm immediately going to try to get back to colder. When we talk about a positive feedback loop, something changed and I'm going to change it more and more and more. I'm essentially going to create a new balance point. We don't want normally your platelets to be forming blood clots. If we form blood clots all the time, all day, every day, that's how you get a stroke in your brain, for example. So we don't normally want platelets to be making a blood clot. But when you have a blood vessel that's torn, we do want to make a blood clot. So we create a new normal, or we create a new homeostasis point where my platelets are all spitting out their proteins. We're building a clot. Once I've accomplished my purpose, once I've built a blood clot, I'll go back to those platelets just floating by. I'll go back to the platelets not spitting out their proteins, not causing me to have a blood clot. Another example of a positive feedback loop is the process of labor. So the way that labor works to birth a child is when the child gets big enough, or when the child drops in the uterus, it puts pressure on the bottom of the uterus, which sends a message up to the brain. And when that message gets to the brain sends a message back to the uterus to contract, to push the baby. Well, as we contract and the baby gets pushed farther, that pressure at the bottom of the uterus is sent back up to the brain. And the brain says, okay, let's contract the uterus again. So we have a positive feedback loop that goes and goes and goes, where the uterus detects something, the brain sends a message to contract the uterus, etc. This goes and goes and goes. Thankfully, not forever, as any women who've had a baby can experience. It's not forever. It's until we reach our new normal, where the baby's born. Then that process will stop. Then there's not the pressure on the uterus once the baby is born. So positive feedback loops, we're not going back to our original normal, we're making a new normal. Whether it's making the uterus contract as our new normal, or whether it's making my platelets split out their proteins. That's my new normal. Until we get to a point where we've addressed the problem, then we'll go back to our regular normal, our regular homeostasis. So question in the chat about the different glands that are involved in some of the feedback loops. That is correct, that the hypothalamus and the pituitary gland are heavily involved in a lot of the different feedback loops in the body. You will discuss that in depth in AMP2. So for the sake of AMP1, we don't need to know which particular parts of the brain or which particular hormones do different things. But for AMP2, you will. That will definitely be a discussion there. I don't remember. Let me see. Hey, so I don't have a slide of it, but at the very end of this packet, there are some review questions about the skin, about the loops, about that kind of stuff. Please don't ignore them, even though we're not doing them in class time here together. That's a really great study tool for you to get you ready for class next week. Friendly reminder that next week we will be back in person, and we will take a quiz that reviews the content here from lesson number 7. So your lesson number 7 homework assignments are due Sunday night to get you ready for the quiz that we're going to take on Monday. Any last minute questions about anything related to the skin? Dusty's good. Oh, y'all almost didn't remind me. Dr. Hollis needs to draw you a penguin. Here, we're going to go all out, and I'm going to give you all the colors. So here we go. While we're thinking of questions, I like to draw what are called macaroni penguins. Macaroni penguins have little feathers coming off the side of their head. So when I was in high school, I like to make them green. I don't know why. So for your viewing pleasure, since you stuck around with me on Wednesday at the end of our class, Dr. Hollis drew you a green penguin. All right. Well, I'm getting no more questions in the chat, so I'm going to go ahead and stop the recording. I will stick around if you have any last minute questions. For my friends who have lab today, plan to meet me at the lab room at about 9:25. I will open up the door then, and we'll get in there, hopefully get started right at 9:30. I suspect, by the way, that it's not going to take you the whole lab time to take that exam. Most of my Monday lab friends finished up in really good time. So I'll see you at 9:25, and we will take that exam and be done with lab for the week.